As we look on to the lambda genetics, we have to learn about first three sets of genes. First one is the immediate early gene. Second one is the delayed early genes. And the third one is the late genes. This is a small structure of the lambda genome, whereas there is a gene called C1 that is located into the middle sides, and there is a N and there is a CRO gene. Now, when we are talking about the immediate early genes, there are mainly two genes that is under the immediate early genes, that is N and the CRO genes. And when we are talking about the delayed early genes, there are many mainly C2, there is C3, alpha, beta, axis, int. There is a lot of them there is present, and for the late genes there are something like the head proteins and the tail proteins of the viral particles and assembly proteins. Now, if we look onto the genetic structures, first genes that is synthesized is the N, and here is the promoter located, which is as it is into the left side, so we will call it PL, and another promoter is located onto the just right side of the C1 gene, that is called the PR. So lambda mostly going through with the lytic cycles or the lysogeny cycle. So if we know about the lytic cycle first, then what is happened? The fast DNA entered the host cells. Then the fast DNA after the enter, this is the genetic setup that is serve as a template for the transcriptions by the host RNA polymerase. So this is very important. They are utilizing the host RNA polymerase to start their transcriptions. So so suppose the host is E. coli. So its RNA polymerase is very specific for the E. coli genome, but not for the lambda genome. So lambda, what they do, they modify the E. coli RNA pol to utilize for their own purpose. Suppose there is a protein named MOT protein that is present into the lambda, which is modified the E. coli RNA pol. Remind it, this should be the E. coli RNA pol. Where it is modified? It's in alpha subunit and the sigma subunit. The sigma subunit is very important. Mainly the 55th amino acids of the alpha subunit and the 76th amino acids into the sigma subunit. After the modifications, of course, these modifications is mainly the ribosylations. They are adding some ribose units into the alpha and sigma subunits. After these modifications, the E. coli RNA polymerase can recognize the viral promoters, that is PL and the PR, but it can't recognize its own promoter. So the viral life cycles now begins with the PL and the PR. After these proteins are seen, after this RNA is made for the N and CRO, these two RNA is made first from the PL and the PR, then comes the translations of these proteins. So here comes the N proteins, and thereafter the delayed early genes and the late gene synthesis. This is a very complicated procedure, so we will describe it later, and they go for the lytic cycles. But in case of lysogeny, after the enter of the phage into the host cells, early gene expressions were blocked by the C1 gene. The C1 protein is basically a 27 kilodalton of protein that will block the early gene expressions. So lambda is in integrated form into the host genomes, that is in E. coli, but they can't go into the lytic phase, so we call it lysogeny. So we will know about the mechanisms of lysogeny first. So starting from the beginning, what is happening? N and the CRO genes, those are the immediate early genes that I am talking about. These genes are transcribed and translated, and there is a termination sequence located here, and there is also a termination sequence located in here. So what is happening? RNA polymerase sit down here, from the PL. RNA polymers sit down here also from the PR, and they synthesize the N and the CRO protein, CRO RNA. So the N protein appears, and it's working. N protein appears, and the RNA polymerase falls out from this area. So beyond this, there is delayed early genes, that is C3 that I mentioned here, and the C2 genes. So C3 and C2 genes never transcribes because there is no promoter here and here. There is no promoter here. So RNA polymerase, after the terminations here, only the N proteins and the CRO proteins appears. So what will happen thereafter? The N proteins appear in our system. N protein appears in our systems, and this is working like an anti-terminator for these termination signals. It is working like an anti-terminator for these termination signals. So if it will interact with the RNA polymerase, and it will modify somehow the RNA polymerase, that we'll know about later. That RNA polymerase can't recognize these termination signals anymore. So they will synthesize now C3 and the C2 proteins. So after the synthesis of C3 and the C2 proteins, these proteins will appear, and they form some dimer. C3 and C2 proteins form dimers that bind at an operator located just downstream of the CRO. Again, I will say this: C3 and C2 proteins that will form a dimer, and they binds at the operator that will first you hear about. So I will draw it again. Here comes the C1, and there is a CRO, and what I say, the downstream of the operator of the CRO. So here is the downstream. This is the downstream. So they will bind here at the operator just located downstream of the CRO. So from the here they start the transcriptions from the another promoter called PRE. So the promoter and the operator sequence are mainly lies here, and here is another promoter termed is PRE. For your reminder, I will draw it again, that PR promoter is from this way, and the PL promoter is that way, and here is the C3 also, and here is a termination sequence, and there is a termination sequence also. So C3 and the C2 dimers binds at the operator just located downstream of the CRO, and here is also a promoter just located into the opposite directions of the PR, that is termed as PRE. This is the full form. What is the full form of the PRE? The promoter repression establishment. As the name suggests, it will repress the whole systems after all. So its name is promoter repression establishment, but its direction just opposite to the PR. So this is the very important. Now the transcription starts from this PRE towards the PR. So the transcription from this when this it starts from the PRE towards the PR, what will happen? The 5' end of this transcript, we know the 5' end is this way, and the 3' end into the backward side. So 5' end of this transcript act as an antisense of CRO. What is the antisense of CRO? It means the when the transcription starts from the PR, it will synthesize CRO in this way. But the transcription when crossed from the PRE, it will synthesize this way. So in that way, if it synthesizes these strands, in case of PRE, it will synthesize the opposite strands. So this is called the antisense. This is called the antisense RNA. So 5' end of this transcript working like an antisense of the CRO. So what will happen? There is a double-stranded RNA that will form from the PR. This RNA will form, and the PRE, this RNA will form. These are the mRNAs from both promoter, and this is from an double-stranded RNA. So the expression of the CRO gene is blocked. Expression of the CRO gene is vital, that we'll know about it later. So the expressions of the CRO is blocked, and the 3' end of this transcript acts as an mRNA of the C1 gene. So here comes the C1 gene. There is no termination signals. So whenever the RNA pol binds into the PRE, it will block the synthesis of the CRO, but it will synthesize 3' end of this mRNA synthesize the C1 gene. So C1 genes first express themselves, and the C1 proteins appears. This is very important that the C1 proteins appears into our system first. So C1 now binds at the operator that is overlapped with a weak promoter called the PRM. So this is the second promoter after the PRE. PRE comes from promoter repression establishment, and the PRM stands for the promoter repression maintenance. So here the C1 gene, and the where is the promoter lies here on these directions, again towards the PRE. Here is the CRO, and if we see about this again, this is the PRE. So 3' end of this transcript produce the C1 protein, and then C1 proteins bind with the overlapping operator here, that is overlapped with the promoter PRM, and this will activate the PRM. So after this activations of the PRM, what will happen? Only C1 mRNA will be produced. From the PRE, we will get the antisense mRNA of the CRO, and thereafter this C1 mRNA. But from the PRM, we will getting strictly the C1 mRNA. So C1 mRNA is produced, and the C1 proteins appears. That's all.